Hi, everybody. Welcome to my recorded lecture. This is for Wednesday, the 3rd of March. I am hoping that we get to where I think we will on Monday. So if I am a little bit ahead, at least you guys will know what we've covered on Monday. So I'm going to start out going to the whiteboard to go over your homework for this week. So for your homework this week, you guys will have your chapter 15 smart book, and that will be due on Monday, the 7th of March. The other thing that I did want to point out, if you haven't been looking ahead very much, is that we do have the following week, the exam 2. This will cover the end of chapter 13. I think we had a couple of sections in there. Chapter 14, chapter 15, and we may have part then of chapter 17. Not a whole bunch of chapter 17, but I imagine a small part of this. And this exam then will open at 5 p.m. And that would be Thursday, I believe it's the 10th of March. And then this will close just like before at 5 p.m. on Saturday, the 12th of March. Again, you'll have 90 minutes. This is an open notes test. We'll talk more about it when I am back, not this week, but the following week, to kind of talk a little bit about that. The last thing that I wanted to remind you of is the library meetings that you guys are supposed to be scheduling. I have those assigned out in an announcement that was sent out, I believe, early last week. And there is a small assignment. It's worth 5 points, and that's just doing a quick summary of your time meeting with Ryan. So those library group meetings, there is a summary due, and that is due, I believe I have it in the week after spring break. So it is due Monday, 3/28. And if you go into week 9, so I'm going to put week 9 is where the assignment actually is. It gives a brief description of what I'm asking you to do. There's a few answers that if you're not quite sure how to summarize it, you can just answer those questions. If you have any questions outside of what I've put up here or on what I've put up here, please do contact I'd like you to contact Alden first, or if you need to, you certainly can reach out to me. Just know that I may not be as quick to return your email in the week after my surgery. But this is what I have set up for you guys this week. A quick reminder that I should be back in lecture on Monday morning, the 7th. We may need to take a couple of breaks during that or the lab, but other than that, I should be back. If I am not, for some reason, there will be an email that goes out and notifies you. Again, though, this is the homework that we have. So from where I believe we will have gotten to on Monday is that we will have completed chapter 14. Chapter 14 was on transcription. We're moving then into translation. So remember, kind of big picture of this, if we have that really, really long chromosome, right, of DNA. So it's this very, very, very long strand. I always like to picture kind of from the floor in my room up through the ceiling. So it's very big. We are specifically looking at just a segment of it of what we would refer to as a gene. And this gene, as we looked in chapter 14, we can do transcription, which is the first step of gene expression or protein synthesis. And from that, we should end up getting, right, an mRNA. We talked about some of the modifications that will happen after transcription on Monday. We know that we have directionality to this, and that this then can go through what is called translation as the second step, right, of this whole process. And this really occurs at the ribosome, right, ribosome. And it's here where our little mRNA will go through the ribosome, that 5' end going first, the 3' end trailing. And what we're going to see is that we're going to have tRNAs that are shuttling in amino acids to this. So I'll write that up, that is a tRNA, and the red here, right, is an amino acid. And we're going to go over how this process works, where as this mRNA is moving through the ribosome is reading it and then bringing in the tRNA that has the specific anticodon to match the codon on the mRNA. And when it does that, it will add that amino acid to a growing chain. And so what we should start to see coming out of the ribosome is these amino acids kind of like beads on a string. And this, again, as I said, is what we would either call gene expression, right? We're expressing that gene. We can also refer to this as protein synthesis, because ultimately the product of this is protein. Okay. So this is, again, just a brief overview. This is what we covered in chapter 14, and we're going to cover this now in chapter 15. Okay, so I am going to jump us back to Blackboard, bring us back to where we hopefully left off on Monday. We'll see if that is true. So I am going to jump us back and bring up our chapter 15 PowerPoint. We are going to start really in the second section of this chapter. I'm not going to go over some of the historical studies that were done in translation. You are welcome to read those, but you are not responsible for those for the exam. So where we're really starting off is on this 15.2, and we're going to be looking at this relationship between the genetic code, we're talking about at DNA, right, and protein synthesis, how that genetic code is transcribed and translated and ultimately can make a protein. So we're going to outline how that information with DNA is used to make the mRNA and a polypeptide. We went into great detail on the DNA to mRNA, but now we're going to really be looking at that polypeptide synthesis. We're going to explain the function of this genetic code, this idea that we have this continuity that flows from the DNA sequence to the mRNA sequence to that anticodon codon between the tRNA and mRNA, and ultimately the polypeptide, the amino acids that are being strung together. We will talk about a few exceptions within the genetic code for what these codons could code for. And then we'll also briefly talk about the levels of protein structure. So once we get this kind of beads on a string coming out of the ribosome, how does this ultimately become a functional protein? And then we'll compare and contrast some of these functions that proteins may have. For most of the time, we think of proteins as often as enzymes. They really help chemical reactions move more quickly and with less energy in a cell, but they can also play a lot of roles in transport and as receptors. So there's a lot of vocabulary here. Hopefully most of this is stuff that you guys are familiar with, even if it's been a while, but we'll go ahead and get started on this. So the genetic code, when we think about that mRNA transcript, we want to think about it as a translation. So it's from one language, which is this nucleic acid language, to the language of proteins, which is amino acids. So in genetics, we think about these nucleotides, and we read them in triplets as that language of the mRNA in the form of a codon. And we translate those then, each of those triplets can signify a one amino acid that will be added onto that protein. And this relies on what we call the genetic code. And so this is a really good example, and this is something that you will have access to. You can access this when you take your exam. You likely will need this for parts of the exam, but this is really looking at the genetic code. And this particular one is showing the codon, so what we would find on the mRNA. And hopefully what you see for this is that most of the codons that we have code for particular amino acids. Some have multiple codons that can code for them, and we'll talk about the importance of that in a minute. Many of them have 4 different codons that you'll still end up with the same amino acid. There are a few that only have 1 or maybe just a couple of codons. We also have a few really important codons that don't actually code for a particular for a particular amino acid when it's found in the middle of that mRNA transcript. We have the AUG that codes for methionine, which is always our start codon. This is the signal to the ribosome that it's going to start translating that transcript into a protein. And then we have 3 stop codons. And this is really the signal to the ribosome that this is the end of that mRNA code that we need to be translating. That is the completed protein. So in order to look at this, when you guys are thinking about codons, one of the things that I do want to point out is that in these ones that have 4, usually 2 to 4 codons that can code for them, one of the things that hopefully you'll notice is that the first 2 locations in that codon are consistent. They don't change. It's the 3rd nucleotide that we tend to see that there is a change. And this is often referred to as wobble. And it was proposed by Watson and Crick that it's kind of a safety net, if you will, in that if you have a mismatched nucleotide here, you still would end up getting the same amino acid in the protein. So even if there is an error or maybe a mutation, we have kind of a safety net to make sure that the protein being made will likely be the right protein. Now, if it's earlier in the codon, in the first 2 positions, it will often be a different amino acid. So with that, we want to think about that genetic information coded within those mRNA in groups of 3. So it's always read in triplets, and those are referred to as a codon. And then this is recognized by the anticodon in tRNA. So I'm going to jump to the whiteboard and just kind of draw out my way of kind of remembering this. So if we look at this, we can think about maybe this is our mRNA. And as I said, it starts at that 5' end and ends in the 3' end going in. And I'm going to do this in a way that it looks like I'm writing this backwards. But if I have my ribosome over to the right, if it's going to be feeding into my ribosome, then it's going to be reading it in kind of that right to left. And so we can think of, I'll just put in some other codons here. And what we want to think about is as the ribosome reads this, it's scanning this mRNA as this is going through here. And it's looking for this first codon because this is the start codon. And this really sets then the reading frame for how it's going to read the rest of the mRNA. And what I mean by that is that AUG is going to correspond to a tRNA that has the complementary base pair, which automatically, because of the base pairing, means that it will have methionine attached to it. And so this is really kind of that translation occurring. And then what we know about this is because that's the start codon, everything after this will be read in these triplets, right? And those will be these codons. So the codons are complementary to the anticodons. And this is really that continuation of kind of that structure and function, the idea that based on the nucleotides and those nitrogenous bases, we can actually tell you just by looking at the start codon or looking at the codon here, what amino acid is going to be added in relation to that. And so that's that specificity between this code, the way that the code works kind of throughout this. I'm going to jump back and look again at the Blackboard at our slides. And this has a little bit different, but hopefully it makes sense looking at what I just drew as well. Up here, it's showing you the DNA, and it's showing a coding strand, which is the top strand. The bottom strand is what is called the template strand. So when the DNA separates at this gene and RNA polymerase can come in and make that transcript, this is actually going to be complementary to the template strand. So T to A, G to C, and so on down the list. Here is our mRNA, and what you can see is on our 5' end, we have some bases, and as you scan it, you should be looking for the AUG starting on the 5' end. And as soon as you find that, we know that's the start codon. that sets the stage then, or the reading frame for all of the other codons downstream. And then ultimately, towards the 3' end, you should run into a stop codon that tells you that that is the end of where we want to translate. And this is just showing you down at the bottom, they draw their tRNAs slightly differently than me, but you can see that we have the complementary, right? A to U, U to A, G to C. And this codes then for methionine. That means that that's the one that's attached to this particular tRNA. Again, we have AGC, which when we look at the complementary is UCG, UCG. That means that serine is going to be the next one. You don't have to memorize these because obviously you will have the table available to you during the exam. But we want to always think about the codons are found on the mRNA, and the anticodons are on that bottom stem loop of the tRNA. And it's actually that anticodon that determines the amino acid that is attached to it. So you can see this continuity, again, as I said, of that code. So there are some special codons. We have AUG, which codes for methionine, but is the start codon. It can later in the sequence also code for methionines within the protein, but the first one kind of on that 5' end is the start codon. We also have at the other end, the stop codons. And there are 3 of those that you would be looking for. When you guys in lab are doing your GEP project, this is part of what you will be doing, is trying to determine the start codon for the given genes that you're looking at and the stop codons, and being able to bring them down to the nucleotide number to be able to map them out basically. The code is what is called degenerate. I often also refer to this as a redundancy, meaning that there are multiple codons that can code for one particular amino acid. We talked about this when we were looking at the chart. In most instances, it is that third base that is the one that could be different and still get the same amino acid. And as I said, that refers to something called the wobble base, meaning that that one is less important than the first 2 bases of a codon. The code is what we call nearly universal, meaning that this is kind of across the board. We see this in all organisms. There are a few exceptions. Please note, though, that I'm not going to spend a lot of time on this. This is not an advanced genetics course really looking at this, but just know that there may be some that have that can code for some other things. So, for example, AUA can code for methionine in mitochondria, and UGA can code for tryptophan in mitochondria. We're not going to worry very much about that. And then there are a couple of others that also code for what are referred to as the 21st and 22nd amino acid. But these are not ones that we really study in this particular class. So this is giving you kind of some examples to the code. As I said, though, this is not one that we're going to spend a lot of time looking at, because you can see that most of these are not organisms that we're really studying in this class. So let's kind of talk about how this polypeptide chain begins to be made, right? We know that the mRNA is being fed through the ribosome in the 5' to 3' direction. The polypeptide synthesis then also has directionality that parallels that. And so during each of those cycles of elongation, when a new peptide is being added, or excuse me, a new amino acid is being added to the peptide chain, there's a peptide bond that will form between those, the last amino acid that was already there and the new one being added. And so you always want to think about the fact that amino acids have one end of them that are called the N-terminus, or the amino terminal end, and that they also have a C-terminus or carboxyl end. So the first amino acid is always going to have that N-terminus or amino terminal open to adding a new amino acid through its C-terminus or its carboxyl terminal. And this is a really good picture of how this looks. So up here with amino acids, we think of amino acids as we look at them. We have this amino end that we think of as the amino end of that polypeptide. We have an amino acid here with its R1 group. We have now another amino group that has added to the carboxyl. It has its R2 group. Again, we have the carboxyl that added to the amino with the R3. And now we have another one coming into the carboxyl end. It has its amino terminal, carboxyl on the other end, And it's this reaction that will form the peptide bond. So the peptide bond will form between the carbon and the nitrogen, and it will give off water in this dehydration reaction. And so this is how it gets added. So the first amino acid that comes off of the ribosome is always going to have its amino group on that end. And then the very last amino acid that we add just before the stop codon will finish in a carboxyl end. So when we think about that, looking at, if we were to think of this as kind of that amino acid, even if we just think of it as kind of those beads on a string, right, that come off of the ribosome, we can imagine that if this one is our first amino acid, and generally we would think of that, right, as methionine as we think of the start codon, this end would be the amino end of our polypeptide. And that means then that at this other end, this would be our carboxyl terminus. And so we always have directionality with our polypeptides as well as with our mRNAs. So with that, I'll bring us back and we'll continue to kind of walk through this. And hopefully most of this is review. I'm hoping I'm not doing too much new. Some of it may be a while ago, so you may have to review it, but hopefully for the most part, this is a review. So as I said, there's always that carboxyl group on that last amino acid that had been added to the polypeptide chain, and that is going to react with the amino group for the new amino acid that's coming in. And then we get this condensation reaction releasing that water molecule. And that you can see there are these peptide bonds then, right, between as we move through these of that C to the high down here, right? We have all of these as we move from that carboxyl end with the amino end. And this is giving you a good view of what this looks like on kind of a little bit larger or kind of a scale that includes both the mRNA with the polypeptide. So again, the 5' is that start. This is showing then that methionine would be the first with its amino terminus, right? Because it's the very first peptide we added. Up here is the R group, right? We have the different R groups for the given amino acids. And then what we see is that each of them ended in a carboxyl that then could have a peptide bond with the amino end of another new amino acid. And this goes down until that very end where we end on a carboxyl terminus. So we want to pause here, or I want to pause here and talk briefly about the structure of proteins. We talked about this a tiny bit at the start of the semester, but I'm going to go over it briefly just to talk about what happens to that beads on a string, if you will, of that primary level of structure of proteins. So I'm going to move us back to the whiteboard. Give you guys whiplash going back and forth. So we're going to think about levels of protein structure. And this falls into kind of these 4 levels. So the first level is what we call primary. And so just as that chain of amino acids is coming off of the ribosome, what you should expect to see is kind of these amino acids all held together by peptide bonds between them. And so it kind of looks like, as I said, beads on a string. And so these are the amino acids, right? And between them is the peptide bonds. But we know that all amino acids have these R groups. These are these groups that vary from amino acid to amino acid, and each of those has a different property, whether it's a positive charge or a negative charge or maybe neutral. And it's those R groups that begin to interact in what is considered the secondary level of structure. And we can have kind of 2 different ways that these could interact. What we can imagine is that for some of them, it will get them to kind of form kind of this, what we refer to as a beta pleated sheet. And it's kind of, they fold back and forth on each other, but it's kind of in this folded kind of way. And so we can call this a beta pleated sheet. And that's just through the way that these amino acids can interact with each other. Maybe two positive R groups can repel each other. A positive or negative may attract themselves. So this is one way that we could get it. The second way that we can get it is that these, it's kind of harder for me to draw when I have my beads, but they can kind of start to form this helix, if you will. So they can kind of start to coil. And so the other type that we can have is what is called an alpha helix. And this is just through the interaction of those R groups. And now not all of this might be in this, right? Some of these, this may just be strung out, some that's not interacting, and same with this as well, right? There may be some that aren't interacting, but this is our secondary. The third one is what is called our tertiary. And I should say the secondary is generally hydrogen bonds. The tertiary tends to be even more stronger bonds that begin to maybe have some of this that is in the helices, some of it is in the beta pleated sheets, and maybe some of it has folded back on itself. And we really think about this as a three-dimensional structure. Again, those are because of the amino acids and the way that their R groups are interacting with each other, this begins to fold itself into it. And if this protein is a protein that only has one polypeptide chain, it could be functional at this point. But many proteins actually need to get to what is called a quaternary level of structure. And this is two or more tertiary polypeptides coming together. If you've ever had me for another class, I always think of this as enzymes, and I always draw this as a Pac-Man, but you think of it as a three-dimensional with maybe an active site. Right. And it's through a bunch of proteins, two or more, that have folded in a way that there's an active site and they've kind of formed this. This is the stage that we typically think of as a functional protein. Okay. So that beads on a string that we started with up top really has to go through these conformational changes before that protein is actually functional and can do its job. Whether it's an enzyme or maybe it's a transport molecule or a receptor, it actually has to fold into these, go through these four different levels usually. Sometimes it can stop at the third, but generally speaking, we think of the quaternary. With that, I'll bring us back, and this is telling you once again that there are these four levels of structures in proteins. And so the protein's primary structure, right, is its amino acid sequence, right? And remember that they each have that side chain, as I said, or R group. And they start out linear in that primary structure, but they're not going to stay that way. There are chaperone proteins that help them start to fold themselves. And this is dictated, as I said, by the properties of the amino acids and the way that they are sequenced within that polypeptide. And so the secondary structure is either that alpha helix or that beta pleated sheet. And that polypeptide can have regions that are primary structure or have these kind of secondary structures as well. And these are stabilized, as I said, by hydrogen bonds. So not super strong bonds, but begin to start this folding process. Then what we start to see is that short regions of that secondary structure in the protein will fold into this three-dimensional tertiary structure. And as I said, this would be the final kind of structure conformation for those that are composed of one polypeptide. But as I said, most proteins are made up of two or more. And so they have to go through what is called a quaternary structure. And this is formed when those different polypeptides associate with each other to form that functional protein. And so this gives you kind of a good idea of this. Starting in that primary structure, it's just the beads on a string, right? Each of these is an amino acid. Then we start to have these hydrogen bonds forming that are either the alpha kind of helices, or we have this beta kind of pleated sheet. And again, this is because of those R groups that are interacting with each other. Then what we see is regions of that secondary structure can begin to fold in a way that makes a three-dimensional structure, and that would be our tertiary. And you can see there's some beta sheets here, and there are also some alpha helices, and we start to see this becoming a three-dimensional structure. And then lastly, we could have more than one of these interacting. again, through those R groups on their amino acids to begin to form this quaternary structure that would be a functional protein. And that's typically what we see happening as this is leaving the ribosome. These things begin to interact. These different chemical properties begin to fold the protein as it's being made. So I'm going to pause here. I'm going to give you guys like 15 seconds to pause, and you guys can go ahead and answer these questions. And then I will come back and answer them. So I would pause the video now and then come back in. All right, so hopefully you took a second, paused the video and wrote down your answers for this. The first question was, the reading frame begins with what? So when we think of the reading frame, I want you to think about the reading frame is always found on the mRNA. And the reading frame is going to begin with what we consider the start codon. And so in this case, it would be AUG, which codes for methionine. The second question is, what are the 3 base units called that are read on the mRNA transcript? So remember, I talked about the fact that we read the mRNA transcript in triplets of nucleotides with their nitrogenous bases. And so we call those codons, right? Those are actually the codons. And then my last question is, what do they match to on the tRNAs? And that's pretty easy, right? That's the anticodons, and those anticodons then dictate what amino acid is attached to the tRNA. So that is really the end of 3.2. We're going to move on to the next one. We still have about 45 minutes of my recorded lecture. So the next thing I really want to talk about is talk a little bit about the structure and function of the tRNA. I always draw it as kind of this upside down clover, and I want to talk about the idea that there's a specificity between that tRNA and a codon in the mRNA, right? We know there's that complementary property between the anticodon and the codon. I also want to talk about some of the key structural features of that tRNA molecule, the anticodon, the stem loops, and where the amino acid attaches. And I'm going to explain how that amino acid attaches itself to the RNA through a special enzyme called the aminoacyl tRNA synthetase. Always make these really big, wordy ones. And I'm also going to talk about how that wobble rule works. Remember, I talked about the fact that that third base may not be as important as the first 2. So I have some vocabulary up here that will hopefully help you understand some of this. So moving forward, one of the things that we know is that in the 1950s, Francis Crick realized that for DNA, if it was going to be coding for the proteins, there had to be something that connected that codon, the codon that was part of that mRNA, to what amino acid was being brought in. And what he proposed in the 1950s was what he called the adapter hypothesis. So basically what he said is there was some molecule, he hadn't identified it, that connects the codon to the amino acid. And it was really Zamsinic and Hoagland who showed the adapter was transfer RNA. They were able to recognize that it was a transfer RNA that had an amino acid attached to it, and that it had an ability to recognize the codons on the mRNA. And that was through this specific anticodon that recognized the codon. And we're going to talk a little bit about that then. And so when we think about this mRNA, right, and we have these codons here, we know that the anticodon then will be complementary that comes in. So if there's a UUC, we expect the tRNA will have the anticodon of AAG. And the fact that this has this anticodon will actually dictate what amino acid could be added to it. So if it has AAG, it will always have phenylalanine that's attached to it. And this is true for all of these. So when we have our codon here of CCG, which is the proline code, we know the anticodon will be GGC. And this GGC will dictate that it's always proline that's attached to that tRNA. And so this is a really important concept, right? That this code follows through, these rules kind of are followed all the way through. And so those tRNAs are actually named according to the amino acid that they carry with them, that they're transporting. And that anticodon, as I said, is anti-parallel to the codon, and it is complementary to that. So one of the things that we'll talk about is those structural features. The tRNAs all have this common structural feature, but there are some things that allow each tRNA to have a different codon, anticodon, and a different amino acid than their neighbor. And that's what allows them to bring in the right one in this code. So the secondary structure of this tRNA, as I said, kind of looks like an upside down cloverleaf pattern. It has these 3 stem loop structures, which means that there are complementary regions of that single stranded tRNA that allow it to bind and make these stem loops. And then there are some variable regions. And the amino acid is actually going to bind to the 3' single stranded region. That's where it's going to be bound to. And then what we can think about is that the tRNAs have our common AUGC nucleotides, but they can also contain some modified nucleotides. And there's up to 60 of these, or excuse me, more than 60 of these that can occur. And these are ways that these tRNAs can differ from one another. So this is a really nice drawing kind of showing you the 5' end of our mRNA. And then it goes down and we have this stem loop where we had some complementary bases that allowed it to form that. Anything that is blue is areas where we could have variability. We have going down then to our other stem loop, and you see we have our anticodon here. And then from this anticodon, we come back up and we have some variable regions, another stem loop up to the 3'. And it's at that 3' region that we have that covalent bond between the tRNA and the mRNA. And so whatever this anticodon is down here, it will dictate what attaches to the 3' region. These variable regions can also play a role in that. So how do they get their amino acids? We can look back here and see that we have this stem loop and we have this open 3' hydroxyl group showing up here. How is it that this one figures out or attaches to the right amino acid that is corresponding to its anticodon? And this happens through a really special enzyme. There's an enzyme that can attach the amino acid that is the right amino acid to the tRNA. And this one is called aminoacyl tRNA synthetase. And the really cool thing is that there are actually 20 of these, one for each amino acid. So each of these aminoacyl tRNA synthetases have a specific amino acid that they're responsible for. So you'll have your methionine one, your proline one. And what they can do is they can do a 2-step reaction where they can add that amino acid to the tRNA and utilize ATP to do this reaction. So basically, these are really important because they're responsible for what is considered the second genetic code. They're in charge of selecting the right amino acid to the right tRNA, because if they attach the wrong amino acid, that means the anticodon that matches up with a codon, if it didn't have the right amino acid, would be adding the wrong one to the protein. And so they are very efficient at this. They have an error rate of less than 1 in every 100,000. And the sequences throughout that tRNA use different parts, especially some of the variable parts, but the anticodon as the recognition site to know which enzyme should be adding the correct amino acid to it. And so those modified bases are also used as markers. And we're going to see briefly what this looks like. So this is kind of cool. I always think it looks like a half moon, but this is the aminoacyl tRNA synthetase. And you can see that it will get a specific amino acid. If this is the methionine synthetase, it will have methionine that can bind here, and only methionine. And it will also bind ATP because it needs that energy to be able to get this reaction to go. And because it has, let's say, methionine here, it is going to bring in the tRNA that has the corresponding anticodon to what would be the codon. And at this point, what we can see is that we get these covalently attached, and then we call this tRNA at this point a charged tRNA. And so the whole job of these synthetases is to charge tRNAs so that they have an amino acid attached to them. Because as translation occurs, those tRNAs are going to give up their amino acid, and they're going to have to go back out and have an amino acid put back on them to continue this equation or this reaction. So that amino acid is attached to that 3' end of our tRNA through an ester bond here. I do want to remind you of the fact that the tRNAs, as we said, that there can be what is called this wobble rule, the idea that the last or the third position of a codon may not be as important as the first two. And there are a couple of exceptions to the rule, but in general, we think of the fact that this third position of the codon is less important than the first two. And so Francis Crick was the one who proposed what is called the wobble rules in 1966. He stated in his that codon anticodon recognition process, that the first two positions strictly pair through that AUGC rule. They have to follow that. But it's the third position that can tolerate some types of mismatches. And so what we know is that there are some isoreceptor tRNAs that can recognize the same codon, meaning even if it's not an exact match at that third spot of the codon and anticodon, they can still bring in the right amino acid. So this is showing this. And the idea of this is that the first two, as we said, have to follow the rule of AU or GC. But the third base pair of the codon can have multiple anticodons that could be here. And these are some kind of modified bases that we're not going to get into in this class, but just know that it has the ability to bind to different ones than we would typically see. And this is kind of the revised wobble rules of the fact that that third base codon, the third base of the mRNA codon, is much less important than the first two, and that's due to what we call the wobble position. I'm going to pause here. This is just some basic homework, or not homework, but basic problem that you guys can solve. So what I would recommend is pausing this here, and then I'll give it 15 seconds or so to give you a chance to pause. And then when you come back, we'll walk through the answers to this. Okay, so hopefully you had a chance to pause and fill this out. So the first question is asking you that, or telling you that this is an mRNA that has the following sequence. And it's asking you first to identify the start codon. So the first thing in my mind that I do is I look for the 5' end. And that's because we're always going to read our mRNA starting at the 5' end. The second thing that I start to look for is what is the start codon. So as I scan down this, I am looking for the AUG, and I found it. So this is what I would say is the start codon. And then it's asking you then to find the complete amino acid sequence that would be translated. So I know first here with this AUG, this is methionine. And then if I go down and I hit GGC from my list, and hopefully I've got this correct, I have that then we would have glycine. I think it's asparagine, then lysine, proline, glycine, can't remember what GLN is. I think it's glutamine, and then a stop codon at the end of that on the 3' end. So please check what you did, go through and see if that ends up being right. But this is a good one to kind of start thinking about the direction that these are. And then the other thing that I would tell you is on that protein, it would be good to be able to recognize which end is the amino terminus and which one is a carboxyl. So the methionine that would be attached here or would have been brought in here because of the codon here would be the amino terminus end. And the glutamine or the stop codon would be then the carboxyl end to our protein. The next question is asking, what is the role of amino-acyl tRNA synthetase? And its whole job is to charge tRNAs with their amino acids. So to add the correct amino acid to a given tRNA. And there's 20 of them, so they're specific for the specific amino acid they add. So the next part of this is the ability of the amino-acyl tRNA synthetases to recognize tRNAs, and sometimes been called the second genetic code. Why has the function of this type of enzyme been described this way? And hopefully it makes sense to you, right? That it's one thing for the anticodon and the codon, so the anticodon on the tRNA, to recognize the mRNA codon. But the second part is that you have to have the right amino acid that has been stuck onto that tRNA, or we're not going to end up with the right protein. And so that's the role of the aminoacyl tRNA synthetase, is that it can actually recognize by bonding with that tRNA, the right amino acid to stick to it. And so that's really the important part, right? That aminoacyl tRNA synthetase, if it has methionine bound to it, it only wants to bind the tRNA that codes for that, right? That has the anticodon that matches that. So that kind of completes, as we've kind of looked through this idea of kind of thinking about some of these different kind of parts of that idea of translation. And we're going to talk a little bit more about the location of where this happens. And the location is going to be at the ribosome. We haven't talked a ton about the ribosome, but ribosomes are found in all cells. They're slightly different in a prokaryote. So in a bacterial cell, they're a different, what we would consider mass than the ribosome that we would see in eukaryotic organisms. But we're really going to start talking about what these look like and how they function with their structure and the way that they assemble in order to get translation to occur. And so I'm going to start out with that idea that that translation is going to occur on the surface of this ribosome. And as I said, bacterial cells have one type of ribosome, and these are called a 70S ribosome, and they're found in the cytoplasm. Eukaryotic cells are a little bit different in that they have two types of ribosomes. They have the one that we're going to be looking at, which is the cellular ribosomes, which are found in the cytoplasm, either loosely in the cytoplasm or attached to the rough endoplasmic reticulum. But they also have ribosomes that are found in some of their organelles. So if it's a eukaryotic cell that has mitochondria or chloroplasts, there are also ribosomes that are found in those that are more similar to the bacterial ribosomes. But we're not going to really look into that very much in this class. We're really concentrating on kind of those cellular ribosomes. And it's important to know that the ribosome is composed of two structures. We have a large subunit and a small subunit. And when these are made, they're actually assembled in the nucleolus of the nucleus in eukaryotic organisms. And they're made up of proteins and rRNAs. And I want to just briefly kind of do a little bit of background on kind of thinking about what a ribosome kind of looks like. So when we think of ribosomes, my pen is going to work now. Let me check this. Sorry, I'm going to double check that might take me a sec to make sure that my pencil is working. Sorry. Let's go back and try that again. Yeah, so with ribosomes can differ by organism. And so if we think about this, all cells have ribosomes, so all cells have them. But we know that cells can be divided into prokaryotes, and this would be bacteria and archaea. And then they can also be divided into eukaryotes. And this would be plants, animals, fungi, all of those good things. So plants, animals, fungi, protists, this is kind of a big group. But they have differences with their ribosomes. So when I think of a prokaryote ribosome, it has two subunits. It has a large subunit and a small subunit. And in prokaryotes, the large subunit is what is called 50S, and the small subunit is 30S. And these are, and Kenzie, you're going to be very proud of me. Kenzie has had to correct me a million times in Intro to Micro, but these are actually called Svenbergs. And these are a centrifugation measurement, meaning that if we were to put these in a centrifuge, the level that they would be found at, if they're separated, is that the large subunit would be found at the 50 Svenberg level in the centrifuge. And the small subunit would be, so I'm going to put this as small, and this is the large, would be found at the 50S. The really weird thing about Svenbergs is that they aren't additive in the way that we normally think of math. And so in bacterial cells, they actually have a 70S ribosome. So when they come together, and we were to centrifuge them, they would be found in the 70S level. I know that can be confusing, but it's always good to keep track of. Eukaryotes have slightly different ribosomes, so they have a large subunit and a small subunit again, so large and small. And in this case, they have a 60S subunit and a 40S subunit. And of course, this does not add up to 100. These are what are called 80S. So we have an 80S ribosome. And again, this has to do more with mass rather than kind of weight or something, which is why it's not additive. But it's actually the coming together of these two that they have to come together for it to be a fully formed ribosome. And you can think about that mRNA in both cases would kind of lay across the smaller subunit, kind of expand, looking up into that larger. And so this is how we would see the mRNA as it kind of snakes through it. Okay. That was a little bit of background on ribosomes. I will bring us back. And please, I do hope that I got that right, Kenzie. If I didn't, you can correct people when we come back. But I think it's Svenberg's. I think I've called it something else forever, but I think I've finally managed to get it. So those are the ribosomes. We're going to talk about how these work as the place where the mRNA is translated into a protein. So as I said, that translation occurs on the surface of that molecule, that macromolecule complex called the ribosome. And I talked about the fact that they have a large and small subunit. So this is giving you some idea, and we're comparing between the bacterial versus the eukaryote. And as I said, it's that sedimentation coefficient where it ends up in kind of that centrifugation. So if we look at the small subunit, we would have a 30S in the bacterial cell and a 40S in the eukaryotic ribosome. The large subunit then would be a 50S to a 60. And as I said, when we add these together, this is the 70S and the 80S. And there are proteins that are also involved in this. This is not just ribosomal RNA. They're made up of ribosomal RNA and proteins. So one of the things that's really cool to look at, this is a really cool photograph of ribosomes in the act of translation. So the ribosomes are all these little blue kind of bubbles along here. The red that is kind of strung through here is the mRNAs. And you can see that this one transcript is actually being translated by multiple ribosomes. And coming off of the ribosomes are these growing polypeptides. And what hopefully you see is that they're kind of longer over here, which means it's transcribed more or translated more of it. Whereas here we're just starting the translation. These ones haven't even had proteins that have emerged. So this is kind of cool. This is called a polyribosome, or sometimes will be shortened to polysomes, which are talking about a transcript that has multiple ribosomes that are translating it simultaneously. And so you can think about that 5' end of the mRNA. As it exits one ribosome, it can go ahead and enter another ribosome and be translated. So these ribosomes are just kind of factories that are reading these mRNAs to make the polypeptide. So as we think about this, during the bacterial, so we're going to start briefly with the bacterial, although we'll also talk about the eukaryotic translation as well. But during bacterial translation, that mRNA is actually going to lie on the surface of the smaller subunit. And as that polypeptide is being synthesized, it's actually exiting through a hole of the large subunit. And ribosomes have these 3 distinct sites. And this is in the large subunit that we find. And this is where the tRNAs will interact with the mRNA. We have what is called the peptidyl site, the aminoacyl site, and the exit site. And I'm going to draw this up. Please have patience with my the way I draw this, but hopefully this makes sense as we think about the ribosome. So again, we're looking at the ribosome. And if I draw it with kind of that small subunit, I like to draw this big subunit with kind of these indentations that there are 3. And I think of this, if I have then kind of the way I think of my directionality is if I have my mRNA stringing through here, we know that the mRNA is going to enter this starting with its 5' end, and the 3' end will be going through last. And the way that we look at this then the way that we think of this as we see this going through is that we can think of this as this particular site is the A site, which is the amino acyl. And this is where most, not all, but most of the tRNAs are going to enter. And then this site, this next kind of opening in it is going to be called the P site. And this is the peptidyl. We often think about this is where the peptide bond is going to be formed between amino acids as they come in. And then the last site is what we refer to as the E site or the exit site. And this is where the uncharged tRNA, once it's given up its amino acid to the growing polypeptide chain, will exit. So this is kind of my brief drawing of what this looks like. We can think about this as we think about those tRNAs, right? And so we know that the tRNAs with their anticodon and their amino acid off of them up here are going to be able to come down into these sites and give up their amino acid before going off, before exiting. And we'll talk about how this happens. We'll both do it on the board and looking at the slides as well. So with that, I'm going to jump us back and think about how this kind of works with these functional sites of ribosomes. And so this is giving you maybe a slightly better drawing of my picture, if you will. But again, we can see in this case that we have our amino acid. And when we look at this, we can see that we have our mRNA stringing through there and getting read by the ribosome. And so as it goes through, what we see is that we can have these anticodons matching up with the codons, and we see each of these little balls up here is one of the amino acids being added to the growing polypeptide chain. And I'm sorry, I had misnumbered mine, so I'm going back just to show you because I had them going through, and I apologize for this. Our 5' should have been over here and our 3' here. I had been drawing it the other way all the way through, but this is really, so this is moving in this direction when we're looking at this, meaning that it started, our 5' started over here and has moved through this way. So I apologize. I'll pause and let you guys kind of check that. This is what happens when you draw your ribosomes in different directions, depending on when you're doing it. But the 5' would have entered through on that side where the A is and eventually coming out along where that exit site is. So hopefully that didn't mess up anybody too badly, but we'll go back and look at this. So again, as this is going through, as this is moving from right to left, the ribosome is going to be looking for that AUG. And when it recognizes the AUG at the P site, it will allow the tRNA with the start anticodon to come in with methionine. And that's really going to be what is called initiation of translation. So I'm going to pause here. We have another checking in. Briefly, I'll pause it, or you guys should pause the video for now, and I'll wait 15 seconds before I come back in and answer these questions. All right, so hopefully you paused and you've answered this. So my questions were, what is the ribosome size of a bacterium? So the ribosomes found in bacteria are 70S. So they have a subunit that is 50 and 30. The ribosome size of a eukaryotic cell then is 80S, and that is the combination of that 60 and 40S subunits. Define polysome. This is where multiple ribosomes are translating one mRNA transcript. So that kind of completes kind of our initial kind of look at kind of ribosomes, the tRNAs, and kind of thinking about what's going to happen. And we're going to get into then, I'm going to at least get started then on the stages of translation. We're going to talk about the 3 stages of translation, which are similar to basically the 3 stages of that original transcription, right? We have initiation, elongation, and termination. And we're going to also kind of compare and contrast bacterial and eukaryotic translation. Obviously, eukaryotic translation tends to be a little bit more complex than we see with bacterial. So as we said, these are the 3 steps, and I'll go to the whiteboard. We can kind of keep track of them here, talking about the 3 steps of translation. So 3 steps of translation. So the first one is going to be initiation, and this is really quite a bit of things are happening in order to get the mRNA oriented right, the ribosome around it, and recognize and looking for that start codon. Elongation is fairly simple, right? It's just that reading of that mRNA, bringing in the correct tRNA when needed. And then that last step is going to be that termination. Basically, when do we stop translating the mRNA, and how does this then complex allow the mRNA to leave, the ribosome to leave, and then that polypeptide to be released from the ribosome? And we're going to go through each of those steps. We likely won't completely finish this, or maybe we will get through most of this, I guess. We'll see how far we get, and then pick up on it the week that I come back. So going back and kind of looking at this, we want to think about, and I know this can be kind of a confusing chart. And so I want to kind of walk through it here, and then we'll go over to the whiteboard probably and walk through this as well. So we're going to start out at what is considered kind of this first step. So we can imagine that there is the large subunit and the small subunit kind of floating around in the cytoplasm. We have charged charged tRNAs that have amino acids attached to them. And then we have our messenger RNA. And you can see here, they're showing you that there's some bases upstream of our start codon. Then there's a very long stretch that is just codons. And then eventually we get to a stop codon with maybe some of the mRNA trailing off afterwards. And so initiation is really going to be the fact that those ribosomal subunits come together right at that kind of start site. So this mRNA is going to interact with a small subunit. And as they're interacting, when we get recognition of the start codon, this large subunit will also be attached. And what we see is as it's scanned, going from 5 prime, so it's scanned all the way down when it hit AUG, which is the start codon, the first tRNA that has the anticodon to that, which has methionine attached to it, will come into the peptidyl site. This is initiation. Now, hopefully you see the E site and the A site are open. And this is where we move into elongation. Initiation is the first amino acid coming in, that methionine, and this connection showing that we've got that start site. What happens from here on out is going to be just a repeat of the exact same steps. What happens is that ribosome will slide along the mRNA, so it will let this mRNA start to go to the left here, and this next codon will be moving in. What happens is when it's at the A site, though, this next codon, is that the second tRNA will come in with an amino acid that is attached, that matches that codon there. What we would have then is we would have a tRNA here with an amino acid and a tRNA here with an amino acid. And we would have an empty E site. What happens is an interaction between the 2 tRNAs in the P and A site, and this first amino acid will be attached to the second amino acid through a peptide bond. That means that the one that is in the P site now is uncharged once it releases its amino acid, and it will move to the exit site. When it moves to the exit site, it will leave the ribosome to go out and be recharged by one of the synthetases. And this will just continue on. Each of the tRNAs in each of these sites will continue to move to the left. And this will continue to happen. And you can see here, this is showing that you have an uncharged tRNA leaving, a tRNA that has the most recently added amino acid with the older ones that are attached to it. It will then transfer this chain to the new amino acid in the A site. Because it then is uncharged, it goes to the E site and leaves, and the one in the A site moves to the P site. And this just repeats itself. it repeats itself until the stop codon comes through, and at the A site, rather than a tRNA with an amino acid coming in, a release factor is what is going to match up to the stop codon. When that comes in, it will cause the ribosome to release the mRNA and the completed polypeptide, and these can be recycled and go back and do this again. So I'm going to do it on my whiteboard. I get quite messy with this. I apologize, but it's kind of the way that I look at it as we start to think about this. And so I kind of want to show, as we think about this, is that we have our ribosome, right, with our 3 sites. That's our large subunit, and we have our small subunit. We can imagine, and we can refer to these. I'm going to say this is the E site, which we talked about earlier. This is the P site, and this is the A site. And we can then draw our mRNA. This is going to be our 5'. You can imagine it going back here to our 3'. And what I'm going to do is I'm going to do the initiation and then kind of look at this going through as we go through. So we may have bases that are up here, right? We may have A, C, G, G, C, who knows? And then we come in, and here we're going to have our AUG. Remember, this is our start codon. And so when this is being read, as this is going from this mRNA is going in here from right to left, when the AUG is recognized, what's going to happen is that a tRNA with an anticodon that matches that is complementary, so we're going to have a UAC comes in, right? This is our tRNA. And attached to it is going to be methionine because that's our start codon, okay? Now, what's going to happen is we're going to have another codon, right? And more codons as they go down kind of this 3' direction. And what can happen is that in this A site, after we get initiation, is we're going to start getting elongation. And what's going to happen is coming into this A site, if we have, and I don't have it off the top of my head, so please ignore the fact that I won't know which amino acid it is that's attached. But then coming into the A site will be that next amino acid carried by the tRNA. And so at this point, we have the E site is empty. The P site has the first tRNA with methionine attached, and our A site has the second tRNA with an amino acid attached. And this is initiation occurred right when that methionine carrying tRNA came in, and now we're at the elongation stage. And what's going to happen is that there is going to be a bond formed between the 2 amino acids, and that methionine is going to be attached to the first one. Excuse me, to the second amino acid. What happens next is this uncharged tRNA, so he's going to move over here. And so that would be the UAC, right? But he's uncharged, right? There's no amino acid attached. So this guy can then exit out. This one will move over into our P site. And so what you'll have is you'll have this amino acid that had, this tRNA that had a GGC that now has an amino acid and methionine attached. And that will open up the A site for a new tRNA to come. And so everything just moves in this way towards that exit site. And this happens until we end up getting to a stop codon, which then will break this whole setup. We'll break it all apart, and the polypeptide will leave. All of these uncharged tRNAs will go out and get new amino acids attached to them, and that mRNA can go on to another ribosome to be translated. So let's go back briefly to our Blackboard, to our slideshow, and kind of refresh on this. But it's really good to kind of think about how this works in real time. And I think, let me see if I have it. I can't remember if I have the video or not attached in here, but what I'd like to do is I'm pretty sure, sorry, I have this video. Double check. Yeah, this is going to be the protein synthesis. I can't remember if we've talked about this before, but this is a good one to show again. And what I will be showing on this, I will pause it at a couple of places to quickly explain what's going on. But in this case, this is a eukaryotic cell doing transcription and translation. But I'm going to pause it when we get to the ribosome. So let's go ahead with this.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix.
So this is the RNA polymerase, right? That's making the mRNA, the messenger RNA.
And copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process.
So I'm pausing it here. And the main reason I'm pausing it here is that if this is the ribosome, right? So the big subunit and small subunit, and this is the mRNA snaking through, we can think of this as the exit site, the E site, this as the P site, and this as the A site. So the A site is where all of the tRNAs come in, except for the first one. And we see that the amino acids are transferred to that tRNA in the A site, and we can see this growing. And as these give up their amino acid, they move into the E site and exit. So hopefully that's clear.
Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. The innermost.
So hopefully that was a helpful kind of dynamic view of what we were just watching, of kind of going back to what we saw here as the steps that occur. I think I am going to actually pause at this slide. So we're going to stop at slide 38. And this is where we will come back on March 7th when I am back in class. And we should be able to finish up chapter 15 quite quickly and then get a good start on chapter 17 on that Monday. If you have any questions about any of this, please do reach out to Alden, or you are welcome to shoot me an email. But otherwise, I will plan on seeing you all. On the Monday after my surgery. Thanks a lot.